Brilliant. Well, hi everyone. Thank you for attending today's webinar, Molecular Bioanalysis Considerations Along the Drug Development Journey. I'm Beatrice Bowlby, and I'll be your host for today's event. Before we introduce you to our speaker, I'd like to quickly cover a few housekeeping items. So firstly, all the information about today's webinar can be found on the right-hand side of your screen, including the Q&A tab. You'll have the opportunity to submit questions to today's presenter by typing your questions into the Q&A box on the side of the window. You may send in your questions at any time during the presentation, and you'll find all of your asked questions in that tab. Please note that other attendees will not be able to see your questions. We'll collect these after the webinar and address them then. If you encounter any technical problems during the webinar, please use the online chat support function, which can be found by clicking on your profile icon, navigating to the help desk, and clicking on the chat with support icon. Alternatively, contact us at clientprojects@tndf.co.uk for further support. An on-demand version of today's webinar will be available following the conclusion of our live event and can be accessed using the same audience link that was sent to you earlier. So we are delighted to have Keith Renier presenting for us today. Keith has more than 13 years of biomedical research and development expertise in PCR, cell-based assays, signal transduction, pathways, and flow cytometry. His research has primarily focused on cellular mechanotransduction, tumor immunology, and cancer biology. He joined LabCorp in 2020 and now serves as the scientific point of contact for internal and external GLP and GCP projects. Thank you again for joining us today, everyone. I'll now hand over to Keith to start today's presentation.
Hello. Yeah, thank you so much for the introduction. Happy to be here, excited to discuss some of the things along the molecular bio pathway and kind of items that, you know, as a SME for the LabCorp group, really tried to develop and help clients kind of move the drug development process forward a little bit more efficiently and effectively. She kind of already went over my contact information and biography. Really, where this presentation stems from is my ability to kind of talk with other teams and groups that are looking for the molecular bio piece and how we can, you know, effectively, you know, move development of a method, validation of a method, and basically translating that into a sample analysis piece to effectively support whatever type of study that you might need for a particular drug product. So my presentation today will look to outline some of those considerations that you might want to keep in the front of mind, have that forward-thinking approach to gain efficiencies at certain locations that we may not typically look to, being so segmented in some of the development basically avenues. It makes it difficult to kind of come together and gain some of these efficiencies that we might not initially think of. So we want to basically define a context of use for planning these studies for that end goal, which is that clinical translation. So I'm going to look at mainly supporting cell and gene therapy, but some of these kind of avenues can be used to support other types of drug development. But here today, I'm going to look at key validation elements and some of the lessons learned along the way in terms of how we've collaborated with other folks and really tried to streamline things as much as possible that might not have been done, you know, early on, but really developed through that whole drug development journey. So some of the objectives that we'll really try to nail down is some of those considerations for packaging and building those methods for preclinical and clinical use. You know, developing that assay context of use, you know, whether it be for a particular model or therapy, you know, what makes sense for what you're using it, you know, what's that endpoint? You know, it may be a different design or product modality that we might need to have some additional context of use, you know, developed into that. And then also, you know, leveraging some of the expertise that we've developed along the way, right? So the PCR field is newer in this kind of cell and gene therapy supporting phase. So the regulations are kind of coming along in that sense. So, you know, what's going to be best for that clinical translation. So like I mentioned, cell and gene therapy is really what we focus most on in terms of the PCR space for our group. Our group is I'll get into that a little bit more, but we're based out of Greenfield, Indiana, and but we support things globally, right? So these cell and gene therapies, they present that new horizon of drug development, right? So like previously things that we weren't able to treat, we're able to target, track, and kind of treat. So this creates a nuanced bioanalytical consideration for some of these therapies. And in many cases, like I said, the regulations do kind of come in behind as that science is developed, right? So really working with that quality lab testing partner will give you kind of a leg up. Someone that's been in the field and worked on some of these safety tests, PK/PD, looking at biomarkers, inclusion, exclusion, and really packaging it for something it's going to be used for, say a submission or eventually in helping a clinical trial and supporting that phase. So as many of you know, these new therapies are very complex structures, and they're getting more and more complex as we move through the different generations of these types of drugs. So really have to think through the heterogeneity of it. What are the testing requirements? What do we need to look at, throughout that drug development process? What's in terms of like safety markers, and like I mentioned, PK/PD. The field is rapidly developing, obviously, both in the manufacturing and the analytical piece. So there's more and more requirements in terms of, what does it take to quantify these products? Are they effective, and how do we characterize that, basically? So we look at that in terms of developing bespoke methods that will, analyze these things downstream, pre and post dose. So like I mentioned, our team is based out of Greenfield, Indiana, but we do have supporting teams across the globe. And I think, like I mentioned, we have a lot of experience in the cell and gene therapy space, but also some of this that in terms of gaining efficiencies may be able to be applied to a lot of different types of drug modalities. So I think keeping that in mind, as well as having that global aspect of things with a unified leadership team, it's really helped us to support a number of different types of these therapies. With aligned technology, aligned SOPs, kind of executing us along the way, it really helps us support that global aspect of pushing a drug through the different phases of a drug development process. And BioA really is that connection of the preclinical to clinical phase that I think that we can pull through a lot more effectively. And that's what we've looked to do with our group and how we work with others as well. Yeah, so in terms of our capabilities, obviously we have a lot of experience with all different types of ATMPs. And typically, the gene and viral therapies that we see and support are those AAVs, lentivirals, and retroviral types of therapies to deliver that product, whether it be the DNA or RNA kind of modality within the vesicle, the delivery vehicle. Another one that we typically support is CAR cells in general. So a lot more immune type cells are being developed to have this CAR structure attached. So we need to have testing in place for those to adequately quantify and kind of track the persistence of the cell, alongside a patient as it goes through, as he or she goes through a clinical trial. So we do have various bespoke methods to these types of drug development products. We have experience in the preclinical regulatory piece. We do have GLP, GCP based testing, and also additional layer of compliance. We do have CLIA, where I'll get into this a little bit later, but really adds that extra layer for patient safety and patient testing where necessary. And I'll discuss this in a little bit more detail. But we utilize the numerous qPCR and ddPCR platforms and really try to work with clients to really understand what their products' intentions are, goals are, and how we can best support that drug development process. Because as you know, there's a lot of different steps. So as mentioned, some of the common therapies, the viral vectors, the CAR immune cells, and what we're seeing a lot more of are the LNPs, the lipid nanoparticles. Basically the packaging to deliver a particular RNA product is what we're typically seeing. But mainly we develop bespoke methods that will target this product. So it would typically be a backbone element in clipping some sort of the delivery or the packaging to ensure that our quantification is specific to what is being dosed, in a tox study or a clinical trial. So, but there are ways, and I will discuss this a little bit later, in terms of how we could use this for more than once, right? Like if you have multiple trials or a pipeline approach that's using a particular vehicle, how can we gain efficiencies there to really effectively push your pipeline forward, but also cut out some of that additional validation, timeline, cost piece is really what we're trying to do in the end game. So I think really looking at like the capabilities of the PCR testing, you hear certain terms in terms of part of whether it be a tox study or a clinical trial. You often hear vector shedding, which is a safety test to evaluate the product and detect the DNA in a biofluid or something that's excreted. So we can track over the course of a trial or a study how much of that particular product is being shed post-dose. Biodistribution is really the uptake of that product. So what is being, how much of the product is there typically in tissue or blood, and then quantifying that. Typically we look at downstream expression. So it'll be a qPCR assay that looks at the DNA, and there'll be an RT-qPCR assay that looks at basically downstream RNA expression of that product or of that delivered vesicle. So, and that kind of leads into that gene expression piece, detecting, a gene of interest or a connected gene that is downstream of what you intended to basically target, so that we can look at, hey, was this effective? Did it do its job? And how much of this do we have versus what we expected? And as you can see on the right here, these are typically the type of a number of assays and the samples that we typically see. So it's very well split in terms of preclinical versus the clinical phase. So carrying through obviously would gain a lot of efficiencies in terms of supporting the preclinical to clinical phase and supporting that translational piece. So that's key for us to really, if we can, gain efficiencies and cut down on things that I mentioned earlier in terms of like the validation, testing, timelines, and being ready for clinical testing. Okay, so for really jumping into the preclinical considerations, how can we translate this to a clinical focused method most effectively? Excuse me. So first, we want to look at, typically we look at, hey, we want to develop a method for this preclinical phase, but what makes sense for your model? So, eventually you want to translate this into a clinical space. We often have this discussion with folks in terms of, what's needed here, qPCR versus ddPCR in a biodistribution or vector shedding analysis study. And it really comes down to what makes sense for your model or your drug modality. If you're typically dosing at very high concentrations, it may change the game in terms of what method you need to develop and how it would best be used in a clinical space. We'll also get into some of these additional validation pieces where we can gain efficiencies, thinking about extraction efficiency, or typically call it like a spike recovery in appropriate matrices that we might use to support a clinical study. Having stability of your product and really understanding not necessarily the product formulation, but how is your product stable in a particular matrix that you tend to test or tend to look at in a clinical study. Are there typical clinical compliances that you might need to look into as you move from the preclinical to clinical space? Typically, how your product is manufactured. Is there anything carried through that might be something that the FDA looks for in terms of, checking a box? And I'll get into that a little bit in more detail. And then I spoke a little bit on the platform methods, but if you have multiple types of these vesicles or delivery vehicles that we can really target and develop a method for, that might pull through into a platform method that we have in the space that we can use for multiple things in your pipeline. So first, really looking at qPCR versus dPCR or ddPCR. So traditionally, I think most would have a more of a quantitative PCR background where you look at relative quantitation, which is basically you create a standard curve and quantify the DNA concentration and back calculate the unknown samples to that basically standard curve. And then in droplet digital PCR, it's more of the newer age tool, provides absolute quantitation. So there's no standard curve needed, really measures the absolute number of molecules present in a particular sample, and really provides that endpoint PCR cycling. And like I mentioned, it's really necessary to determine the end goal, what makes sense for your model, not only for IND submission, but as we translate to the clinical phase and for clinical reporting, how's that best going to look? So that's what we would have that early discussion on. So for your typical, this is a representative, basically standard curve for a qPCR assay, typically have 5 to 7 levels of that standard. We incorporate all the necessary controls. So like a no template control to ensure that there's no contamination, as well as an extraction negative control that, controls that extraction piece upstream of the qPCR analysis. We do look at basically what's called a spike control within each sample to ensure that the sample is not inhibited. So if we do get a negative or a 0 or undetermined reading, that it's a true undetermined reading versus something that was carried through the extraction process or was in piece of the part of the qPCR assay preparation. Typically, we can analyze up to 18 samples per plate, incorporating that spike replicate for inhibition. We use it for multiplexing, obviously, identifying typically use it to support a lot of the AAV assays that are really high dosing levels. Our dynamic range for this qPCR assay is a little bit larger, so we can look at the vector shedding and biodistribution that way pretty effectively. And we do get good sensitivity on the bottom end as well. So it's not necessarily antiquated in that sense. We do get typically 50 copies per microgram. And then for vector shedding, typically go down to 25 copies per microgram of DNA loaded. Now for the ddPCR piece, like I said, there's no standard curve, but we do typically incorporate 3 to 4 quality control samples that kind of span that dynamic range of the limit of quantification for that assay. And then with those controls, we can establish that threshold of what's positive versus negative, and quantify the number of typically droplets or PCR positive droplets for that target, a little bit more space on the plate without that standard curve. And once again, we can multiplex a number of different ways, whether it be gating on amplitude, kind of similar to a flow cytometry assay. But typically we do a duplex assay that looks at vector copy number, gene expression, gene editing, and obviously the CAR-T monitoring as well. So in terms of benefits and limitations of the ddPCR, very good precision and accuracy. Signal to noise is reduced, much cleaner in that sense. It removes the standard curve, so it creates more space there. Not as much of a concern for inhibitors due to basically the drop it partitioning into its own reaction. It detects very small fold differences in target in between the samples, and we can use obviously a very low concentration of starting material. Limitations, as I mentioned, the dynamic range is much smaller. Really need to optimize how much of how much DNA or product we can load, and the window that we can look at that is really more focused on the lower end. Whereas if you have something coming in much higher, typically in the ranges of you would see in a like an AAV designed therapy, sample dilution would be necessary, where we'd have to kind of retest those samples to make sure that they're in the dynamic range for a ddPCR assay. And then moving on from the ddPCR versus qPCR discussion, additional elements of the validation that we typically look at for supporting a preclinical phase, we would look at extraction efficiency or a methodology versus for whatever species we typically want to assess. For the preclinical phase, we try to fold in all the matrices. So we want to understand the extraction methodology process, that workflow that we plan to support in sample analysis. So we'll do a spike recovery in those pre-extraction, and then assess those to develop a method bespoke to the product. So it really depends on your study design. And I think this is where we can pull through the clinical piece if we incorporate, obviously, that translational to the clinical space. If there are human matrices that we plan to assess, we can validate those in concert with the preclinical pieces. And that really gives us a leg up in terms of having that method developed and ready to go for that clinical translation, as your core validation will not change. So the method will be in place to begin, the first patient in kind of testing. So we really need to align with a clinical trial. It may be in a stage that's not as developed, but really, incorporating some things that, we're going to need to, assess in the clinical space. We already know. It may be blood or a different matrix that's going to be assessed in the clinical trial for shedding. It could be a FFPE or a fresh frozen biopsy tissue that we would need to basically validate and ensure that we're robust and reproducible in terms of our extraction methodology. And then just fold that into the initial validation to save time and cost, so we don't have to jump back in line to validate something new when we could have done it initially. Another piece that commonly thought is an afterthought in terms of bioanalysis testing is stability of the test material. So we typically want to characterize how your product will be or how it will act in a actual matrix that we plan to test in the clinical space. Oftentimes in the preclinical phase, we will do what's kind of called a batch analysis. So it's really a short window that will turn samples around versus a clinical trial, which could take 1 to 5 years. So you really need to understand, as things are being collected at a collection site, what is the turnaround time needed for your test material? So if we can understand that ahead of testing, we have that that validation and that method in place for testing said samples really puts us in a good place to begin the clinical trial and the clinical sample analysis pretty quickly without having to jump back in and waste a lot of time on revalidating some things. So we really need to assess sample type, what will align with the clinical trial, kind of similar to what I mentioned in the extraction efficiency piece. That will be the similar. So we would execute the core validation and then bring in the stability piece kind of in parallel. So as, say, a preclinical trial is being assessed for sample analysis, we can run these stability assessments in the particular matrices that we need to look at. This will fold into the original validation. We can initiate things earlier for the preclinical testing, and it could cut down on the amount of stability we actually need to look at if we have this done ahead of time. So we might not have to go out as far, and we can understand a scheme that we can batch samples more appropriately. So the overall stability study can help determine that batching and how we can efficiently turn samples around quicker, or if it gives us some bit of a gap to really wait for maybe maximizing plate space, or kind of cutting down on the number of assays needed. So that stability data can establish that prior to first patient in aligning with that clinical trial. Another piece, not clearly understood all the time at the preclinical phase, but having early discussions with the regulatory agencies can really help determine this. Like I said, the clinical trial protocols may not be developed at a stage that you're really ready to execute, but it may give you some cues in terms of what's needed in terms of endpoint determination and how we describe that in a clinical trial protocol. So the clinical compliance consideration is really key to get ahead of kind of what's needed for that translational piece. Something that we really should be thinking of early on. And I think it gives us a heads up and a leg up in terms of, if there are potential safety methods that we need to have in place, we can get those started as soon as possible, and may not be something that we look at initially in the preclinical phase. Excuse me. And as I mentioned, the CLIA and IVDR, the additional layer of testing compliance, really need to understand if the sample analysis for a particular bioanalysis assay, will that be used to make patient decisions? And that's where you really need to fold in the CLIA IVDR piece and really understand that early on, because it may be that you need to validate to certain qualifications and characterize your method appropriately. And that's something definitely with the regulators in terms of developing your clinical trial program that you would really need to establish early on, and we can fold that into a validation as well, and fold in the right folks to make sure that we're compliant where we need to be compliant. One of those things that often comes up for us is the safety piece, the retroviral, replication competent lentiviral, retroviral, and also in the human herpes virus types of clinical safety testing that are needed and need to be established to go alongside, say a vector copy number or a different type of bespoke PCR assay, that samples will be collected in parallel for your clinical trial. And it really comes down to, how are these things manufactured? What are some elements in the RCL, RCR, HHV that could be carried through and potentially harm patients? So it's a safety method that needs to be in place, and something that I think we could fold into the validation as well. And also, I'll discuss a little bit later in terms of a platform method for said testing, so that it can not only be established, but it can be established to the compliance needed, such as CLIA. And oftentimes, this may be a timing thing, and you don't want it to be an afterthought. Really need that regulatory feedback as soon as possible. And I know they're happy to discuss things as early as possible. And I know really need to understand that piece of how it's going to translate. So happy to have those discussions early on and really build that piece into the validation if we can. Mentioned a little bit in terms of platform methods. So if there's thinking about a drug development pipeline, oftentimes groups have, they may be different payloads that they're carrying, but the packaging is very similar. So if there's a way to conserve the target amplicon between those, we can develop a bespoke method that will really carry us through not only preclinical to clinical, but study to study, modality to modality. So we can develop those pieces in mind as a molecular bioanalysis team to really identify a conserved region. It kind of comes into number 2 in terms of the methods used for similar cell and gene therapy. So the safety pieces that I just mentioned, obviously the replication competent lentiviral packaging and envelope elements are conserved across a number of different transduction types of manufacturing pathways. So that's something that we can, and we have developed a platform method to identify that envelope protein for the RCL, ensure that it's not present in patient samples, and use that across a number of different clinical trials. Typically, we would just bridge to a group's basically RCL packaging plasmid or kind of viral makeup, and then we can utilize the in-house method to test those for clinical trials. Another one that typically comes up is, as we see more and more allogeneic types of cell therapies and how those are sourced, potentially brings the human herpes virus prevalence in the human population into the ballgame. Basically, there's a potential risk for those immunocompetent or incompetent patients to basically have this HHV element introduced through the allogenic cell therapy. So that's something that we need to monitor as well. And the prevalence could initiate basically a medical treatment or something in that nature. So that's typically a CLIA approach for that would be appropriate as well. So next, I'll jump into basically a comprehensive, product support portfolio. We've had a collaboration with a client for over 9 years now in supporting a number of their AAV gene replacement therapies. We started early on with them in terms of developing a method of per molecule, and there were multiple different validation studies executed. But we tried to, as I mentioned, lessons learned, fold in some of these elements I've discussed in terms of conserving an amplicon across the pipeline. So we developed a bespoke method for analysis of the biodistribution in multiple animal models, and also folded in some of the human matrices to really be ready to translate that quicker. Obviously it wasn't there early on, but as we developed more and more understanding of what's needed, we could fold that into their subsequent validations, support the preclinical phase, but also have a method in place along with stability elements to understand a batching scheme that will best support the clinical phases 1 through 3 and up to product release and long-term follow-up. So in total, we have 4 different molecules with the group. We still support them to this day. There's, it mentions here over 19,000 samples analyzed. I mean, that's growing as we move along, but we helped release the particular products into the field, and also have these methods standing and specific to a packaging vehicle that if they do continue this pipeline, we can support those as well. So it's really in a good place to incorporate a lot of the efficiencies that I discussed. And looking to do this more and more with other groups. We start to fold some of these elements in, have those early discussions, and it really is going to put us all in a good place in terms of summary. I'll go through that a little bit, but having those in place in terms of, you know, what's best for your model. Obviously, that was what we first discussed, and often have that discussion in terms of qPCR versus dPCR, ddPCR, and happy to have those discussions as well. And it may be a case where a group does have a method standing internally, and we need to translate that, and happy to do that as well. And that could also gain efficiencies to where we can cut down on what's needed regulatory-wise, including QA oversight and all those different things. Mentioned the extraction efficiency piece. Want to pull through, obviously, the preclinical matrices and multiple species that we may support there, but also fold in the human elements to have a method standing for clinical translation. Understanding the stability piece is key. How is our product performing in a matrix? And will that, you know, support a batching scheme that's outlined in a clinical trial? Understanding the clinical compliance. A lot of the safety pieces that may be needed in terms of, you know, differences between the preclinical versus the clinical trial space, and having those methods standing to help support clinical trial testing. Folded into platform methods, can we gain efficiencies by developing bespoke methods to a particular conserved region, or have a safety test in place ahead of the clinical translational side, and have that validated and ready to go, can be utilized as a platform method for a particular product or modality as well. And the number 6 is really the key one, and it really is, it comes down to communication, and really having it early on, whether that be with the regulators, in terms of, you know, what's this translation look like. Groups are often segmented in terms of early development, preclinical phase, and then they have different clinical teams, and those teams don't necessarily communicate as much as they may need to get some of this kind of in place early on. So I think early communication, both internal and external, are key to really fold a lot of these pieces in and have that understanding early on. It may be a case where you don't really understand what the clinical phase is going to look like, but at least discussing that and having elements in place, it can begin to develop a game plan for some of these said deficiencies. So with that, I think that's all I have. Yes, thank you.
Excellent. Thank you so much. Yeah, thanks for that presentation, Keith. If you do have any questions, Keith, then please continue to submit them using the Q&A button at the side of your window. We will now start addressing some of your questions. So to begin, what is ideally critical to best incorporate some of the efficiencies discussed?
Yeah, like, so I said, the communication piece is key, right? Not only having that with the bioanalysis testing teams, but also internally with both the preclinical and clinical teams as well. I know there's often, obviously it takes a lot to get things going in both phases. So the early communication is key, but also if you can identify some things that can be carried through, such as a conserved amplicon region, can help with testing and getting things set up early on.
Brilliant. Thank you. So as the field continues to evolve, what do you see coming forward for PCR and its capabilities in terms of new modalities?
That's a good one. In terms of, I think for us, we're seeing a lot more ask for how do we quantify small RNAs, and it's very tricky in terms of the PCR space. So we're seeing an ask for not only downstream expression of maybe delivering a microRNA or a small interfering RNA to knock something out. We're also seeing quantification of those, like how much of something do we have present, where is that out at in the biodistribution or shedding space. So I think it's coming up more and more. And I think we're really trying to develop those to align with the guidelines and white papers to date in terms of context of use and moving those forward as well. So that's a big one.
Brilliant. And then as a subject matter expert for your group, what are some of the biggest challenges you encounter when translating bioanalytical methods from the preclinical to the clinical space?
Yeah, like I said, it would probably be the segmented groups and getting everyone on the same page. I think that's everyone typically has a job to do, right? So they're focused on in the moment, but getting the buy-in from both sides to really, especially early on. I mean, it can take a lot of time to add in some additional elements when you're trying to hit timelines, right? So timelines are a piece of that, but getting buy-in from both groups across a team's pipeline and things like that, I think that's key. It's very difficult to do, especially trying to hit budgets and things like that to make things work.
Excellent, thank you. And then the next one, in your experience, when is CLIA compliance necessary from the molecular bioanalysis perspective in supporting a clinical trial or phase?
Yeah, that's a tricky one, a little bit of a gray area in terms of that piece, often discussed with multiple QA departments. But our stance is typically, if there are medical decisions to be made from the bioanalysis testing piece, so if your results are going to be used to be, to treat a patient or make medical decisions in that front, the CLIA or even more is needed in that sense. So that's typically how we fold that in. And we do look to get feedback from the regulators early on that piece, but that's typically our stance is if there's going to be medical decisions to be made.
Sure. Okay. And then when thinking about product stability, when would you ideally build in those assessments to keep on track for patient sample testing?
So typically we would fold those in after we've established a core validation. So we would run through our qPCR assessments, establish those methods, and establish the extraction efficiency piece, and then jump into stability. So ideally we have some time there as we're going through that to understand the clinical or the human matrices that we might need to look at. So I think really want to fold that in at the end of a core validation piece, and we could definitely have those discussions early on. But really, it may be a case where you don't understand your clinical trial protocol completely at that point, but we can have those discussions and really identify maybe there's a couple matrices that we can set up early on, and it may be a case of, oh, well, we want to add this, and we can fold that in as well. So it's really after that validation, the core validation, we would build that in.
Thank you.
Mm-hmm.
Okay, then we've got another one. Any thoughts on dPCR versus ddPCR for supporting this type of work? Are you seeing any trends for either technology?
Yeah, so in terms of we are bringing online more, so a dPCR platform, and so that's often a discussion as well, is can we translate, say, a qPCR method to the dPCR space, and we have that capability. What does that take? It really depends on the product and the context of use, but we are seeing more of that. I think the dPCR platform does present more of a higher throughput for us. We're excited about that, and that will be utilized more in the preclinical space, so you get that bolus of samples. It's able to be used in that front a little bit more effectively, whereas the ddPCR has been more of a clinical use at this time. But we are doing some internal innovation pieces to look at those three in concert and really understand what's needed for each, and are they translatable, and it may be a case where you need to do that bridging for a particular product or test material.
Brilliant. Thank you.
Mhm.
And then we've got, so they're saying a topic of whole blood stability is being discussed in industry now, but it's not applicable in ligand binding assays. So what do you think about how qPCR and ddPCR would be applied in those types of assays?
Yeah, blood is tricky, and oftentimes can cause PCR inhibition, right? So I think understanding that piece for us, especially if it's going to be in you're testing human blood samples, we need to understand that turnaround time, because if our collection is not being effectively executed, sample is degrading or something within the blood, and we're not getting anything downstream, we really need to understand, if we can understand that earlier on in the validation, we can get some answers into, hey, we might need to change the statement of work in the clinical trial collection piece. So if we can look at that early on, that would be helpful as well. But blood can be tricky for PCR related elements. So I think that it would be kind of crucial for us in that sense.
Okay, thank you very much. Thank you for answering our audience questions. Before we wrap up, do you have any last comments you'd like to add?
Just that the field is exploding right now, and the PCR space is also, kind of along there with it. We're excited to, work with folks on that piece and in developing context of use and pushing these guidelines forward. So us at LabCorp are excited about the opportunities ahead. So thank you for your time, and I really appreciate it.
Excellent. Okay, well, I would like to once again thank our wonderful presenter, Keith, as well as you, our listeners, for your time and questions. You'll receive an email with how to watch this webinar on demand shortly. Don't forget to visit us at bioanalysis-zone.com for more webinars. Thank you again for attending. We'll see you soon.